To begin the tutorial, we need to start VisANT, which is available from the button at the top right corner of the website. This will launch a Java applet, which will allow us to load networks and interact with them. VisANT can also be used as a standalone Java application if you need to work without web access. The first step of the tutorial is to clear the network, so that we can load one of our own choosing. We're going to turn off AutoFit, as it says in the instructions, and make sure that we are looking for interactions from the yeast database. Start by searching for the gene STE4. This will give us a diagram showing all of the interacting genes. Hovering the mouse over the edges displays a list of methods providing evidence for that particular interaction. Now we're going to expand one of the interacting genes to take a closer look by double-clicking on it. This will perform another query to find all of the interaction partners for that gene. We can do this recursively, to query the interaction database in a graphically intuitive manner. Some simple mouse operations can be used to pan and zoom. Using the right mouse button, I can click and drag the canvas to recenter it. I can also use the mouse wheel to zoom in and out. Double-clicking will also recollapse the nodes so that you can trace back to where you started. Before we walk through step 3, let me describe a scenario to help illustrate how the features we are introducing could be useful. Suppose that DNA JA1 and RPL28 are genes that are both found to be differentially expressed in a hypothetical gene expression study, and we would like to know if their protein products share any interaction partners that would help explain the observations of our study. DNA JA1 is a heat shock protein with a C-terminal zinc finger domain, and RPL28 is a ribosomal protein with both RNA and protein binding functions, so biologically, it is not clear how the two proteins might be related. In the following steps, we will use VisANT to query the interaction database for ways in which these two proteins might interact. It is easy to determine that the two do not interact directly simply by adding one of them to a new network and looking for the other among the neighboring proteins, as we saw previously. How could we find out if the two proteins might interact with a third common protein? We can look for such a direct neighbor interaction between the two proteins easily by building a simple network of all the proteins known to interact directly with DNA JA1 and RPL28 individually, and checking to see if those networks of neighbors are connected. When we try that approach, we will find that the two proteins do not share a direct neighbor interaction. Perhaps the two proteins still share a pathway, however. To explore this possibility, we expand the network we just built once more to add in all proteins that interact with those first-level interactions, and then look for paths between the two proteins within this dense network. Finally, in step 4, we take a look at building an ad hoc network using data we provide to VisANT and see how to query for interactions without expanding the network using data from the database. Okay, now we're at step 3. We're going to clear the network view and we're going to start over. This time we do want to select AutoFit, and we're going to look for genes under Homo sapiens instead of yeast. We're going to build a little bit of a network from a list of genes, starting with DNA JA1 and RPL28. If we click Search, we'll see a small network of interactions. These are the first-level interactions between both of those nodes. The default view shows the genes we searched on encircled by all of their interaction partners. We can do a layout of this network to get a better idea of how these two nodes are connected to each other by doing Layout, Spring Embedded Relaxing. Running that for just a few seconds makes it clear that these are two disconnected networks, at least for the first level interactions. Now we want to select all nodes and go under the Nodes menu and say Query Interaction of selected nodes. This will expand our connections by using the database. Now we want to select the original two nodes that we had searched for. Hitting the search button again, we'll just do that. And we want to find the shortest path that connects them in this newly connected network. If we look at topology, shortest path, find shortest paths, this will give us a list of paths that are tied for the shortest distance between these two genes. In this case, DNA JA1 and RPL28 are 3 nodes apart from each other in about 14 different ways. Now we are going to do step 4. This again starts with another new network. We're going to be searching the Homo sapiens database of interactions again, and we're going to start with the same 2 nodes. But instead of using the search field, we're going to list the genes down here in the Your Data field. This will allow us to create an ad hoc network of our own. I'll add a couple more. And clicking Add creates a small network. You can see RPL28, DNA JA1 are there, and P53 and SMAD4 are there, connected by an edge because of the way I added them to the network. All these nodes are already selected in the viewer, and if we say Nodes, Query Internal Interactions, this shows us all the edges between those nodes that are found in the database. We now see a new connection between RPL28 and SMAD4. That's it for the first tutorial. Thank you for checking us out. VisANT is being continually refined, and we have several upcoming features under active development. You can now predict the function of network modules using gene ontology term enrichment analysis, also known as GOT. Also available is a new network module enrichment analysis feature, also known as NMEA, that allows you to detect network modules or pathways whose expression values are correlated with phenotypic differences, such as case and control groups. Our lab has also been studying functional linkage networks, which integrate data from multiple sources to allow you to predict the function of previously unknown proteins or modules in the network, and to explore correlations between network modules. We have been applying biological networks to the study of human diseases, exploring ways to detect network modules based on network structure, studying transcriptional networks in various organisms, and researching algorithms for network alignment across multiple organisms. So, thank you again for visiting. Come to visant.bu.edu for more information about our lab, tutorials, and examples on how to use VisANT.